This video will be discussing more about 4 subtopics. Firstly, the effect of postsynaptic potential, which is neural integration. Secondly is autoreceptors, then, the third is other types of synapse. And the last one is nonsynaptic chemical communication. Let's begin. With the first subtopic is the effect of postsynaptic potential, neural integration. What is neural integration? Neural integration is the interaction of the effects of excitatory and inhibitory on a particular neuron. This picture shows the structure of neuron. As you can see, there are several structures like myelin sheath, cell body, axon and dendrite. These excitatory or postsynaptic potential, or also known as EPSPS, which represented in red, are then transmitted by means of passive cable properties down the dendrites, across the soma, to the axon helix located at the base of the axon. If the depolarization is still strong enough, when it reaches this point, the action potential will be occur at axon. Okay, let's reconsider what would happen if at the same time, the several inhibitory synapses also become active. Inhibitory postsynaptic potential, also known as IPSPS, are hyperpolarizing, and they will bring the membrane potential away from the threshold of excitation. Thus, the IPSPS they produce, which shown in blue, will diminish the size of EPSPS and prevent the occurrence of action potential at axon. Next, we move to the autoreceptor. What is autoreceptors? Autoreceptors can be defined as a site on a neuron that binds the neurotransmitter released by that neuron, which then regulates the neuron's activity. Autoreceptors can be located in any part of the cell membrane. For example, in the dendrites, the cell body, the axon, or the axon terminals. Thus, when it is stimulated by molecule of neurotransmitter, the autoreceptors do not produce changes in the membrane potential of the terminal button. Instead, they function to control internal processes, including synthesis and release of neurotransmitter. Also, autoreceptors are usually G-protein coupled receptors, which is slow response in EG via a second messenger. Now, we move to the third part, which is other types of synapses. Basically, as we can see on the picture, there are several types of synapse, including axosomatic, axdendritic and also axoxenic synapse. Axosomatic or axdendric are the synapses that frequently been referred to the effect of postsynaptic excitation and inhibition. But axoxenic synapses work differently, where they alter the amount of neurotransmitter released by terminal buttons of the presynaptic axon. And the last part that we'll be discussing today is nonsynaptic chemical communication. In this subtopic, nonsynaptic chemical communication will be focused on neuromodulators and hormones.
For neuromodulators are chemical released by neurons that travel farther and are dispersed more widely than neurotransmitter. Mostly are peptides, chain of amino acids that are linked together by chemical attachments, called peptide bond. Then, neuromodulators are secreted in larger amount and diffuse for longer distances, modulating the activity of many neurons in a particular part of the brain.
As you can see, this is a figure of neuromodulators. Next, hormone are secreted by cells of endocrine glands, or by cells located in various organs. It can affect the activity of cells that contain specialized receptors, located either on the surface of their membrane or deep within nuclei. Cells that contain receptors for a particular hormone are referred to as Target cells. Many neurons contain hormone receptors, and hormones are able to affect behavior by stimulating the receptors and changing the activity of these neurons. This figure shows an action of steroid hormones. Steroid hormones affect their target cells by means of specialized receptors in the nucleus. Once a receptor binds with a molecule of a steroid hormone, it causes genetic mechanism to initiate protein synthesis. Because steroid hormones are soluble in lipids, they pass easily through the cell membrane. They travel to the nucleus, where they attach themselves to receptors located there. The receptors, stimulated by the hormone, then direct the machinery of the cell to alter its production.